Good morning, everyone. Today, we'll be discussing genetically modified potatoes. Let's get started. A genetically modified potato is a potato that is modified by genetic engineering for pest resistance, tweaking the amounts of certain chemicals produced by the plant, and to prevent browning or bruising of the tubers. Varieties modified to produce large amounts of starches may be approved for industrial use only, not for food. 1. Currently marketed varieties of GM potato used for food. Innate varieties. The genetically modified innate potato was approved by the United States Department of Agriculture in 2014 and the USDA in 2015. The cultivar was developed by J.R. Simplot Company. It is designed to resist black spot bruising, scratch, browning, and to contain less of the amino acid asparagine that turns into acrylamide during the frying of potatoes. Acrylamide is a probable human carcinogen, so reduced levels of it in fried potato foods is desirable. The innate name comes from the fact that this variety does not contain any genetic material from other species. The genes used are innate to potatoes and uses RNA interference to switch off gene. Move on next slide. The innate potato is not a single cultivar. Rather, it is a group of potato varieties that have had the same genetic alterations applied using the same process. Ranger Russet, Russet Burbank, and Atlantic potatoes are innate versions of the potato varieties. 2. Previously marketed varieties used for food. New leaf. The new leaf variety of potato was designed to resist attack from the Colorado potato beetle due to the insertion of Bt toxin-producing genes from the bacterium Bacillus thuringiensis. The insect-resistant potatoes found only a small market. Used in industry. Amphora, also known as A92-527-1, was a cultivar developed for production of pure amylopectin starch for processing into waxy potato starch. Move on next slide. Pre-marketed varieties. A modified Desiree potato was developed in the 1990s by biochemist John Gatehouse at Cambridge Agricultural Genetics, later renamed Axis Genetics, to express the Galanthus nivelis agglutinin (GNA) gene from the Galanthus snowdrop plant, which caused them to produce GNA lectin protein that is toxic to some insects. This variety of potatoes is the one which was involved in the Pustai affair. Second point is, in 2014, a team of British scientists developed modified version of the Desiree cultivar that can resist infection after exposure to late blight, one of the most serious diseases of potatoes. They developed this potato for blight resistance by inserting a gene, RPI-VNT1, into the DNA of Desiree potatoes. This gene, which conferred the resistance to blight, was isolated from a wild relative of potatoes, Solnum vinderi, which is a native of South America.